Proteins play a pivotal role in the proper functioning of any organism. The synthesis of proteins occurs in a cell in two phases, namely transcription and translation. The process of converting mRNA codon sequences into an amino acid polypeptide chain is called translation. Prior to translation, mRNA, which is synthesized in the nucleus of a eukaryote cell, migrates through the nuclear pore into the cytoplasm of the cell. The cytoplasm also contains tRNA, amino acids, and ribosomes, all of which are involved in the process of translation. The amino acids and tRNAs in cytoplasm become activated by a process called the charging or amino acylation. During this process, amino acid binds with ATP in the presence of amino acyl RNA synthetase and is activated into amino acyl AMP enzyme complex. This complex reacts with tRNA and transfers the amino acid to it. As a result, tRNA becomes activated as amino acyl tRNA and the enzyme and AMP are liberated. The tRNA has an anticodon loop which determines the amino acid that will link to it. This anticodon of tRNA is complementary to the codon sequence on the mRNA and will only bind to that codon. Apart from tRNA, a ribosome is another component required for translation. It is made up of structural RNAs and about 80 different proteins. In the inactive state, a ribosome consists of two subunits, a large subunit and a small subunit. The large subunit consists of two sites, the P or peptidal site, also called the donor site, and A or amino acyl site, also called the acceptor site. At these sites, the two subsequent amino acyl tRNAs bind and facilitate the formation of a peptide bond between the two amino acids. The ribosome also acts as a catalyst in the formation of the peptide bond. Another component in translation is mRNA, which consists of the start codon AUG that codes for methionine, the stop codon, which is either UAA, UAG, or UGA, a region that codes for the polypeptides, and regions that are not translated into proteins called untranslated regions or UTR. These regions are present at both the 5' end before the start codon and the 3' end after the stop codon and ensure efficient translation. Translation occurs in three phases: initiation, elongation, and termination. In the initiation phase, the small subunit of the ribosome moves along the mRNA strand to locate and bind to the start codon AUG. The first amino acyl tRNA carrying the amino acid methionine with an anticodon UAC binds to the start codon at P site, after which the large ribosomal subunit binds to the small ribosomal subunit containing the mRNA. Another amino acyl tRNA now binds to the A site of the large ribosomal subunit, and the first peptide bond is formed between these amino acids. The peptide bond formation is catalyzed by the peptidyl transferase enzyme. The formation of this bond requires energy, which is obtained from ATP. Simultaneously, the first peptidyl tRNA gets removed, leaving the amino acid. The next phase is elongation, in which the ribosome runs along the mRNA in the 5' to 3' direction, moving from codon to codon and adding subsequent amino acids to the growing polypeptide chain. This is caused by the elongation factor translocase, and this process is called translocation step. On reaching the stop codon, UAA, a release factor or RF, binds to it, terminating the translation and releasing the polypeptide chain from the ribosome into cytoplasm. After the termination, the small and large subunits of the ribosome dissociate and the mRNA degenerates. The polypeptide chain is therefore an end product of the translation process that takes place in the cytoplasm.